Hello and welcome to this Introduction to Cell Biology video. I'm Dr. Aled Roberts, and in this video we're going to introduce the concept of the human microbiome, exploring the differences between endogenous and exogenous microorganisms, how they interact with the human body, and the roles they play in both health and disease. Now, when we talk about the microbiome, it's important to consider several key aspects and definitions. The first is the microbiota. This refers to the community of microorganisms that inhabit a specific environment. It includes all the different bacterial, fungal, archaeal, and protozoal species, as well as non-living entities such as viruses and bacteriophage. Now, the definition of the microbiota can be either broad or specific depending on the context. For example, we can talk about the human microbiota as a whole. That's the entire collection of microorganisms that live on and in the human body. Within this, though, we can narrow our focus to particular sites, such as the gut microbiota, which plays a vital role in digestion, immunity, and overall health, or the skin microbiota, which helps to protect us from pathogens and maintain the integrity of our skin barrier. Now, in most cases, when we discuss the microbiome in detail, we're particularly interested in the human microbiota as it has such a significant impact on health and disease. However, microbiota are not limited to humans. There are also environmental microbiota, so microbial communities found in soil, water, and air. For example, different soil types can host very different microbial populations, and even within the same soil type, microorganisms can vary dramatically between regions due to differences in temperature, moisture, and nutrient availability. Now, it's also important to recognise that microbiota and microbiome are not exactly the same thing. The microbiota refers specifically to the collection of microorganisms present in a given environment. The microbiome, however, is a broader term. It encompasses not only the organisms within the environment, but also their collective genetic material and the interactions that occur between them and their environment. In other words, the microbiome includes both the who and the what they do. That said, in much of the literature and even in everyday scientific discussions, the term microbiome has become the de facto word used to describe these microbial communities. You'll often see it used interchangeably with microbiota, and that's perfectly fine in most contexts. Now, the human microbiota is defined as the complete collection of microorganisms that live on and within the human body. And whilst the exact species and strains within the microbiota will vary between individuals, the general types or groups of microorganisms tends to remain fairly stable. In other words, two people might not share the same bacterial species, but they'll often share similar kinds of microbial communities performing comparable roles. And so it has been estimated that the human body hosts around 1,000 different bacterial species, representing roughly 30 to 40 trillion microbial cells, though the exact number depends on the source of the data. And when we compare that to the human body itself, a single species, Homo sapiens, made up of around 28 to 37 trillion human cells, it's clear that microorganisms make up a huge part of who we are. And these microbial cells inhabit and help form a variety of microenvironments throughout the body, shaping our health, metabolism, and immunity. So in many ways, we're only half human. We live in a symbiotic relationship with our microorganisms, a partnership that's essential for our development, survival, and well-being. Now, when we shift our focus from the microbiota to the microbiome, we see an even bigger difference. You see, the human genome contains around 20,000 to 25,000 genes, a relatively modest number when you consider the complexity of the human body. But the microorganisms that make up our microbiota collectively contribute an estimated 2 to 20 million genes. That means for every single human gene, there could be anywhere from 100 to 1,000 microbial genes, vastly expanding our functional potential. Now, these microbial genes perform a range of essential functions that our own genome simply can't do alone. They help us digest food, synthesize vitamins, and train our immune system. There's also growing evidence that they can influence our mood and behaviour, linking gut health to mental well-being. And so, when we consider all of this together, Humans and their microbiome can be viewed as a kind of supraorganism, a combined system where both human and microbial components work in harmony to maintain life. Now, when we talk about the human microbiota, we may describe these microorganisms as endogenous microorganisms, meaning that they are naturally found within specific, defined environments on our body. When the body is in a healthy state, these communities are referred to as the normal microbiota. They exist in a balanced relationship with the host, contributing to health and preventing the overgrowth of harmful species. And it's important to remember that the composition of the normal microbiota varies depending on the body site. Now, some of the most heavily colonised sites on the human body include the oral cavity, skin, urogenital tract, and the gastrointestinal tract. And so starting with the oral cavity, this includes the mouth, teeth, tongue, and gums. It's a warm, moist environment that provides plenty of surfaces for microorganisms to attach to, as well as a steady supply of nutrients from food that we ingest. And because of this, it supports a rich and diverse microbial community. Next, we have the skin, which covers around 2 square meters and is one of the most varied microbial habitats on the body. Different areas of the skin provide very different conditions. Some are dry, others moist, and some are oily, and each of these supports distinct microbial communities. The composition of the skin microbiota can also be influenced by factors like hormones, which affect oil production, and in turn, which microorganisms can thrive in certain areas. Now, the urogenital tract also shows a lot of variation, particularly between males and females. In females, the microbiota is dominated by bacterial species which help maintain an acidic environment that protects against infection, whilst in males, the microbiota tends to be less dense and more variable, reflecting the differences in anatomy and physiology between the two. And finally, we have the gastrointestinal tract, which is by far the most heavily colonised site in the body, with a surface area of around 300 square metres. And as we know, conditions change dramatically along its length. It starts with a low pH and high oxygen environment in the stomach and upper intestines, and gradually transitions to a higher pH and completely oxygen-free environment in the large intestines. These changing conditions create a series of distinct microbial niches supporting a huge diversity of microorganisms adapted to each region. And so if we look at the species present in each of these niches, the oral cavity is home to around 750 different microbial species containing a mix of both aerobic and anaerobic microorganisms, including genera such as Pasturacea, Prevotella, Veillonella and Neisseria, and among these are various Streptococcus species, which are the most common and play a major role in maintaining good oral health. The skin hosts around 1,000 different microbial species, with the main groups found on the skin including Streptococcus, Corynebacterium and Propionibacterium, now partly reclassified as Cutibacterium. Now, our Propionibacterium species are the most common and particularly abundant in oily areas of the skin like the face and back, where they play a key role in maintaining skin health. The urogenital tract contains hundreds of microbial species, with some of the most common including Lactobacillus, Prevotella and Gardnerella species. Now, the composition of this microbiota can change significantly with gender, age and hormonal status. For example, in females, the vaginal microbiota before puberty is often dominated by Staphylococcus, Streptococcus and Escherichia species. After puberty, however, as estrogen levels start to rise, Lactobacillus species become dominant, producing lactic acid, which helps maintain that low pH which protects against infection. Now, interestingly, this is a key example in science where the female body has been more extensively studied than the male body, providing valuable insights into microbial influences on things like reproductive health. And finally, we have the gastrointestinal tract, which is the most densely populated microbial environment in the body, with over 1,000 different species identified and a mix of both anaerobes and aerobes, including genera such as Bacteroidetes, Helicobacter, Enterococcus, Escherichia and Klebsiella species. Now, among these, bacteroidetes species are the most common, with gram-negative bacteria playing a key role in digestion, metabolism, and immune regulation. Now, while some areas of the body are highly colonised by microorganisms, others are considered to be sterile. And this essentially means they don't normally contain any endogenous microorganisms. These include areas such as the eyes, brain, circulatory system, lower respiratory tract, liver, and kidneys. Now, this sterility is usually maintained by physical barriers such as tight epithelial layers, protective membranes, and immune surveillance that prevents microbial entry. However, if microorganisms do manage to breach these barriers, the resulting inflammation can interfere with vital functions in these organs, often leading to serious consequences for the host. And so, depending on the body site, some areas are heavily colonised, whilst others have no colonisation at all. This leads us to a few key questions. Do these endogenous microorganisms actually cause disease? With so many living in and on us, is that what makes us sick? The short answer is no. Most of the time, these microorganisms live with us quite peacefully and even provide very essential benefits. The long answer, however, is possibly. Normally, our microbiota live with us in balance, but certain conditions can tip that balance and allow some microorganisms to become opportunistic pathogens. Now, this can happen for several reasons. One key factor is in immunocompromised hosts. When the immune system is weakened by illness, medication, or stress, it can no longer keep these microorganisms in check. Sometimes infection occurs in a different host or body site. A microorganism that's harmless in one location, like the gut, can cause serious disease if it ends up somewhere else, such as the bloodstream or urinary tract. Poor hygiene can also contribute, allowing normal flora to overgrow or spread to places they don't belong. Another emerging factor is acquired virulence, when microorganisms gain new genes such as those encoding toxins or resistance factors that makes them more capable of causing harm in a particular host. And finally, when physical barriers are broken through wounds, surgery, or medical devices, microorganisms that are normally contained can gain access to deeper tissues and trigger infection there. So that's our endogenous microorganisms, or at least a small snapshot of them. Now let's turn our attention to the exogenous microorganisms, and essentially these are everything that isn't endogenous. Basically, they are the microorganisms that are introduced from the outside that don't normally live on or within us, and are not part of the normal microbiota. So where do they come from? Well, there are a range of different sources, and one of the major sources is other people. What's endogenous to me, part of my normal microbiota, might be exogenous to you. Transmission can happen through simple everyday interactions like sneezing, poor hand hygiene followed by a handshake, or even sharing personal items such as cosmetics or towels. Another key source is the healthcare environment. Hospitals and clinics bring together a high concentration of infectious individuals and a high concentration of immunocompromised patients, creating the ideal conditions for microorganisms to spread. Animals are another important source of exogenous microorganisms. These can be both domestic animals like pets and livestock, and wild animals that we might encounter less frequently. Microorganisms can also be acquired through agriculture, from animals that are alive, for example, through direct contact or handling, or from those that are dead, such as during the processing or consumption of animal products. And finally, we have aquatic environments, which can also be important sources of exogenous microorganisms. These can include air conditioners, showers, and taps, where microorganisms can thrive in biofilms and be released in water droplets or mist. Even still or stagnant water can become a breeding ground for bacteria, which can then spread through aerosols and cause respiratory infections. So now that we've explored where these microorganisms come from, let's look at some specific examples of our exogenous microorganisms. So let's look at some examples. From other people, we have things like Staphylococcus aureus, and about 1/3 of people carry this bacterium as part of their normal microbiota, but it can easily spread to others and cause infections, ranging from minor skin issues to more serious systemic disease. In healthcare settings, enteric bacteria are a major concern. They can cause diarrheal diseases, and worryingly, many strains are now antibiotic resistant, making them much harder to treat. From agriculture, a good example is the foot and mouth disease virus, which spreads through direct contact with infected animals or their saliva, milk, or feces. Non-agricultural animals can also transmit microorganisms such as dermatophytes, which are a group of fungi that can cause ringworm, or the protozoan Toxoplasma gondii, which can be particularly dangerous during pregnancy as it can harm the developing fetus. And finally, from aquatic environments, we have Cryptosporidium parvum, another protozoan that causes diarrheal disease. It's often spread through contaminated water, including swimming pools, where it can survive standard chlorination. And with that, we come to the end of this video. Hopefully you found the content useful, informative, and most importantly, easy to understand. Thanks for watching, and I hope you have a great day.